Good afternoon or good morning, whichever applies. I'm Dr. Neil Drew, a health program specialist here at NIH, working for the NIH Countermeasures Against Chemical Threats program. I'm joined today by Dr. Shardell Spriggs and Dr. Jodie Fleming. Welcome to this webinar where we will talk about the current funding opportunities that support therapeutics, discovery research, and early stage development research. As we get started, I'd like to go over a few housekeeping items. We are recording this webinar. Afterwards, we will share the recording and the slides with all the registrants. We are taking questions. You can enter your questions in the chat box at any time. Some of them, depending on the question, some of them will address in the flow of the presentation. Otherwise, we're going to address all other questions at the end of the slides. Specific questions about your proposed research project may be done on a one-on-one basis with staff at a later time. Feel free to email us to set that up. Our emails are included in this slide presentation. Today, we are going to cover a few things. We'll briefly go over the NIH CounterACT program, and then we'll go over the two trans-NIH funding opportunities that we offer, the R21 and the UG3, UH3. And finally, we'll share some tips that we hope are helpful as you prepare your applications. We are going to cover programmatic and review aspects from the funding opportunities, but at a high level. And that's why we have one representative from program and review, Dr. Spriggs and Dr. Fleming, respectively. And we also want to leave as much time as possible to address your questions. So we will go more quickly over some of the background material. I do encourage you to go to our website. You can see the URL and it's the link at the bottom of the slide. It has a lot of information there, including background of the program and particular events that we run, like this webinar. You can also find links to past events, including past webinars that cover some of today's material in more depth. Finally, there are some resources posted that may be helpful, such as the scientific program contacts, information for applicants, and examples of milestones. With that, I'd like to pass this over to Dr. Shardell Spriggs, Program Director in the NIH CounterACT Program.
Thanks, Neil. So the NIH Countermeasures Against Chemical Threats program is a congressionally mandated program that was put in place around 2006 to enhance the nation's preparedness for mass casualty events after the events surrounding 9/11. The mission is to understand the fundamental mechanisms of toxicity caused by chemical threats and the application of this knowledge to develop promising therapeutics for reducing mortality and morbidity caused by these threats. Now, we do this with a focus on exposures that occur in civilian mass casualty situations. This is when many people need to be treated at once. And this is in direct opposition to the warfighter, where you may be able to offer pre-treatments. So our therapeutics need to be fast-acting, easy to administer by first responders that could be encumbered by personal protective equipment. And then our models have to take the CONOPS in use, or the concept of use. So we really stress models that mimic efficacy in these delayed treatment models. And of course, the overall end goal is to deliver better therapeutics as handoffs to advanced development that will most benefit the civilian population. Next slide, please. So chemical threats are toxic compounds that could be intentionally used, such as in a terrorist attack, by governments for warfare, or even self-harm. But there are also accidental exposures that may occur. This can be related to industrial production, storage, or shipping. And so we develop therapeutics for all of these scenarios. Next slide, please. So CounterACT funds research on over 200 chemicals of interest as identified by the Department of Homeland Security. So we have a list that we have to fund these chemicals on. If your chemical of threat that you're proposing research on is none of the list, then we are not allowed to fund it. And so we're really asking you to contact us before you start developing an application. We're happy to talk to you. We would love to have the conversation. As a program, we group these chemicals into toxidromes, such that chemicals within a toxidrome have similar modes of action and toxic effects. And so we have some examples listed here for each toxidrome that we have. Now, grouping this by toxidromes offers better harmony and syncs better across the many government programs that focus on medical countermeasures, including those for advanced development. Looking at these by toxidromes allows you to develop therapeutics based on symptoms rather than actual chemical threat. And the end therapeutic is usually has a more broad, efficacious spectrum. Can we go to the next slide, please? So as you can imagine, a wealth of knowledge is needed to run such a diverse program. So this is a trans-NIH effort that we leveraged expertise across all NIH ICs. The ICs shown here hold grants within the programs, and oversight is by the NIAID, who also runs the larger CCRP program that CounterACT is a part of. On a later slide, within here, and also if you check the NOFOs, we will have scientific context listed for each of these ICs. And again, please don't hesitate to reach out. Can we have the next slide, please? So the CounterACT funding opportunities are designed to feed into one another and promote a progressive translational program structure, beginning with our now basic research mechanisms. So each IC has a basic research NOFO on the street that aligns with the research interest in that toxidrome. And then there are the developmental R21 grants that we have, and then the UG3, UH3 cooperative agreements. And that's really what we're going to focus on today. There's also a NOFO that is an exposure core NOFO shown up in purple, and NIEHS, NEI, and NINDS have signed on to that. One click, please. Okay, so again, highlighting what we're going to focus on, and then we can go to the next slide. So again, we know sometimes it's hard to decide which NOFO your research fits best in. And so this slide is just outlining that for you a little bit. You want to come into the R01 if you're just studying basic disease pathology after chemical threat exposure. So just the progression of the disease, you're not sure of a molecular target, you're looking for maybe some biomarkers. Here, you're not developing a therapeutic, and that's the real difference. But I urge you also to check these specific NOFOs because each one is very different from the others. Now, when you talk about the R21, this is our greatest, I guess, NOFO with level of diversity. So you can do almost anything in here. But it's really only, it's 2 years for limited support. We'll go into this a little bit deeper on later slides. But if you have a candidate but are just looking to develop that preliminary efficacy data to be more competitive in our UO1 cooperative agreement, I said UO1, I meant UG3, UH3 programs, you want to come into the R21. Now, our latest stage NOFO is the UG3, UH3, and that's really what we're going to focus on today. So coming into this, you want to have a better idea of what you're trying to do. You want to have leads that you need to down select from, and that's where you're going to do all of that work. And these are 5-year programs. May I have the next slide, please? All right, can we go back? Okay, so looking at the R21, we said the goal is to generate this preliminary data to get ready for the cooperative agreement mechanism. It's important that with this one, we're really supporting translational research. So everything that you're proposing in here has to lead to a therapeutic that is going to better an outcome for an exposure victim by a chemical threat. And the scope is really broad. We've listed a few here from the NOFO. So creation and validation of clinically relevant in vivo or in vitro models of post-exposure lethality. And so this includes things like 3D organoid models. And we're interested in serious near and long-term chronic morbidities. So we want these single exposure events, but everything that happens after that is fair game for study. So when a standard of care is available, we want to see combinations of new therapeutics that are improvements over the standard of care. We're interested in artificial intelligence and machine learning models for the purpose of investigating and discovering new medical countermeasure strategies, discovery of candidate therapeutics using.
Have your attention, please. We'll be conducting a fire alarm test. I repeat, we are only conducting a fire alarm test, and this is only a test. Please do not evacuate. This is only a test of the fire alarm system. Thank you.
Discovery of candidate therapeutics using primary and secondary screening. This is including high throughput screens. And so again, this is important here to note that you want to do this stuff in the R21. When we get to the UG3, UH3 slides, we don't want you to screen libraries looking at 1,000 different compounds. So that's bucketed here for the R21. Preclinical development, and then of course, in all of our NOFOs, we're interested in
It's not that loud. You can probably talk over that.
okay. FDA approved drugs for repurposing. May I have the next slide, please?
There has been a fire emergency
Okay,
reported.
maybe not if it talks.
While this is being verified, please leave the building by the nearest exit or exit stairway.
He's gonna do.
Do not use
Okay.
elevators.
So if you can't hear me, let me know and I'll stop. For the R21, there are non-responsive applications will not be reviewed. Okay, what this means is that if we get your
May I
application,
have your attention, please?
we're going to look through
May I
it.
have
If your application matches with any of the bulleted items that we're going to go through,
While this
we
is
will
being
have to
verified,
withdraw
please leave the
your
building by the nearest
application.
exit
And
or
so the
exit
first thing that we
stairway.
really want to hammer home is that we have a list of chemical threats that we're allowed to fund. We are not allowed to fund your application if it's not on the list, the current Department of Homeland Security list of chemicals of concern. Best way to avoid this is to check with us. You can click next,
May
Neil.
I have your attention?
Okay, again, our target audience is to develop therapeutics for the civilian population. So we really need the CONOPS or the concept of use for your therapeutic to be amenable during or after this mass casualty scenario. So anything that has to be administered pre-exposure or within the first 15 to 30 minutes exposure within the R21 is not within scope. And again, the one difference here are for ultra potent opioids. We know these are truly fast acting, and it's a really early stage area of research. So for the R21, administration as short as 5 minutes post exposure is important. And there are a few other differences among the toxidromes. So again, we put it here, but the most important thing is that you check with the scientific contact for the toxidrome that you're interested in. Could you hit next, please? Again, we talked a little bit about this. We want that acute exposure. So if your model is for a worker in an industrial plant who's been exposed Monday through Friday, one day, one time a day, that's not us. Okay, but if your model is one exposure on Friday, that fits within our model. So it's one exposure. That's what we mean by acute. But then again, we are interested in chronic effects, meaning long-lasting effects, effects that can last 6 months longer or as short as 3 days after. We hit the next slide, Neil.
May I have your attention?
Okay, we don't do
May I
decon,
have your attention?
or detection strategies. So this may fit better in the SBIR. If you have a therapeutic for this, please just reach out to us. And one more.
Do not use.
Okay,
elevators.
please, all of the research that is for basic disease pathology after exposure, that belongs in the basic NOFOs. This is only for therapeutic development. And Matt, the next slide. Okay, the R21, we just had a receipt date in May. Those are going to review in December, and we will award those in March after taking them to January and February council. Direct cost and project period are standard. If you click the button, you can see our next receipt date. Our next receipt date is May 30th, 2024. And next slide. So one quick consideration is that we only have one time a year receipt dates for the R21 and the UG3, UH3. So let's say you applied this May, you get your award next March. You have to look to come into that UG3 after a little more than 1.5 years of funding. And that's just something we want to outline for you. If you click the button, yes. So make sure that your goals of that R21 are aligned with the entry criteria for the UG3. Just be really mindful of what you're proposing to do based on the information that you have and that next step of the UG3. So please read the UG3, even if you're applying for an R21, so that you can propose and craft your science to give you the best UG3 application possible. And following this will also give you the shortest gap of not being funded by our program. And next slide, please. Okay, getting into the UG3, UH3. The goal here really is everything you need to come down to that lead compound in that UG3 phase, and then in the UH3 phase, optimizing that compound. And when we're talking about optimizing or what we think your end product should look like, that's this well-characterized therapeutic where we want to see affinity, potency, target selectivity, minimal off-target effects, in vivo efficacy, and tox in an animal model that is aligned with our programmatic goals. So that means the concept of use. When you're in the UG3, you really want to see that delayed treatment model. So time for someone to have exposure, for exposure to be recognized, 911 to be called, get on the scene, and then be able to easily administer your therapeutic. That's all important in this late stage NOFO for our program. And then that's also, I think that covers scope too. So we can go to the next slide. Entry criteria, rigorous data supporting your hypothesis. We want you to have lead compounds and assays that you're going to use to down select in hand. We don't want to see you spending 1 to 2 years developing assays or screening large libraries to get that initial hit. You can do some SAR here, and we also want you to have freedom to operate. So get that licensing, work early and often with your tech transfer office. The budgets are a little different for the phases. UG3, you can request up to $350K direct cost. And in the UH3 phase, it's a little more money because the studies are more advanced. These are 5-year projects. So you can, at your option, your choice, divide that up. You can do either 2 years or 3 years in the UG3, and then put the remaining in the UH3. And that's really going to depend on what you already have in hand and the kinds of studies you're proposing to do. The next receipt date is fast approaching, which is why we really wanted to have this webinar right now to go over some of the things that we want to make sure you hit on. So October 17th is that next receipt date. Next slide, please. Okay, now, what's special about the UG3, UH3? Similar to our previous cooperative agreement programs, we have milestones here. What's different here is that we've gated the 2 phases of your project with a milestone. So again, we've talked about this being 5 years. The UG3 phase must be less than 3 years. You can propose 2 or 3 years in that phase. Don't exceed 350K in direct cost. And then the UH3 phase also cannot exceed 3 years. Your choice, 2 or 3 years, with whatever's remaining in your 5 years is here for your optimization studies, 450K direct cost. What's important is that you must submit this as a single application. So when you submit your application, it should have all 5 years of research in there. You need to clearly delineate what you're going to do in the UG3 phase and what you're going to do in the UH3 phase. And that needs to be clear in the specific aims. And then milestones are annual. So if you have a 5-year project, you need to have milestones for year 1, milestones for year 2. If you've only proposed 2 years in UG3, then you need to have a transition milestone before you go into the UH3 phase. Okay, and then year 3 in the UH3 phase, you need to have milestones. 4 in the UH3 phase, you need to have milestones. Now, you might ask, how do you get from UG3 to UH3? And the answer to that is that this is going to be by internal review. Those of you who've been in our program for a while know we had a U01 lead ID and then a U01 optimization. And you had to come back in and submit with that once-a-year receipt date and hope your project received a good score. What this allows us to do is review your research internally and move the project forward into that UH3 phase without you having to resubmit and go through review. We're hoping that this speeds along moving products from early to later stage translational research. But that is why it is imperative that you submit a complete application. So transition is not automatic. We will look at that and move you from UG3 to UH3. And most importantly, we say we want to make sure we say this 6 times. I don't know how many times we've already said it, but you need annual milestones and a transition milestone. Let me go to the next slide. Okay, this is in a NOFO. The big take-home message here is that UG3 phase activities, it can be everything you need to down select to that 1 to 2 lead compounds. If you have some in vitro activities you need to do to look at efficacy, and then some in vivo work to just say, this is my lead. You might have to do a little SAR here on your molecule. You can do that, but everything you need to do to get down to 1 to 2. And then we have the internal review where you've shown efficacy. So Neil, can you click once? I'm not sure. Okay, yeah, we put a little flare on that to get your attention. Okay, internal review, we need that transition milestone. And I'm really harping on this a little bit because we had to withdraw an application for not having transitional milestones. And we had to withdraw an application that last round for not having any milestones. So transition milestone is needed. Your transitional milestone should focus on showing that your therapeutic is efficacious in a delayed treatment animal model, whatever that means to you. And you can call us and we can work on that. And then Neil, can you hit next? Again, UH3 phase, everything around optimization, formulation, developing that TPP falls into that category. Next slide, please. All right, milestones. Milestones need to be clear and quantitative outcomes that set go and no-go criteria. This is going to be very different than what people are used to doing in the R01. Okay, your milestone should not say, I'm going to test 500 compounds. That's not a milestone. Your milestone should say, I'm going to find, 1 compound that does X, Y, Z. And we've put an example here. So test if drug X is efficacious in mice. That's a task, and that's not a milestone. Your milestone needs to be tied to a quantitative outcome. That means you need to benchmark it. Your milestone should have numbers in it. Okay, if you don't have numbers, that tells you right there it's not a milestone. Let's go to what a good milestone looks like. Demonstrate efficacy in reducing lethality when drug X is administered at 20 minutes after a cyanide challenge. And then you have a criteria for success. And your criteria for success is going to be your benchmark that you need to hit to go forward. And here we say you want a reduction in lethality by a certain percentage. That means it's an improvement when you administer it in a method that's amenable to mass casualty delivery intramuscularly here, 20 minutes following this exposure. And then you're letting us know when you're going to evaluate it. And we like to see the rationale of why you're doing this. And again, there are plenty of examples, good examples on our website and on other websites here at NINDS. NIAID may have a few, but please check our website first. The link will be in the slides that are sent out. And there's also a link on our website. You have the next slide, Neil. Okay, again, some of these we can skip through because they are redundant for what's on the R21. So that chemicals of concern list, we're bound by that. Your threat has to be on that list. Check with us. It has to be in line with our programmatic priorities, which is treating the civilian population. Single acute exposure, but we are interested in both short-term and long-term effects after that single acute exposure. We have to have those milestones. If you want help with them, reach out to us. They have to be quantitative. You have to have them for each year. And then also for the transition. And all of that needs to come in as one package. Okay. We don't want projects, and you can't do this anyway, probably because of budgetary reasons, but really out of scope activities are those advanced development activities. And this is really, they're listed in NOFO, centered around the GLP, IND enabling studies, these pivotal efficacy studies in animals, current GMP practices, and then also clinical trials are out of scope. So you should already have your lead therapeutic compound coming into this NOFO. So if you're looking for discovery, that's done in the R21. You can have a couple you're going to down select from, but you need to have a better idea of what you're doing for this advanced NOFO. No high throughput screening of thousands of compounds here. Again, if you have, even 25 compounds, 30 compounds, that can be doable in 6 months to a year that you're down selecting from. That's possible to justify here, but not large scale screening activities. And then also, you need to have your animal models in place. If you want to develop animal models, that's also the R21. And then also going back 2 bullets to, you need to have your assays in place to do that down selection. Okay, so you don't want to spend a year or 2 developing assays to see if your product is efficacious, please, or to identify your products. Please come in for an R21, get all of this in place. It's really going to make your cooperative agreement UG3, UH3 score higher or better. And then again, diagnostics or devices, we are interested in those. We just cannot fund those. What we do is we partner with SBIR and we get those funded through SBIR. Oh, great. At this time, I am going to hand it over to Jodie. If you guys don't know Jodie, she's the scientific review officer that handles the special emphasis panels for the CounterACT program. She ran the last several steps, and so she knows what your applications look like, and she's going to help make them better. Thanks, Jodie, for joining us.
Great, thank you. Hello, everyone. Again, I'm Jodie Fleming, and so I'll just be here to go through the application-specific aspects that you need to consider so that you get the best review possible. And so we have both NIH-specific as well as CounterACT-specific angles that we need to make sure that you are fully aware of and the reviewers are expecting to see. All right, and so to start off with, for NIH-specific, you always must remember that you have to address the enhancing reproducibility through rigor and transparency, right? Implementing rigor and transparency. And you can see the notices here, and those links will be available to you. To break it down into user-friendly words here, what we're talking about here for the rigor of prior research, what you need to include in that application in both either the significance or the background, right? The rigor of prior research, what we're asking for is that you show us, you know, the literature, you know where the holes in that literature are, where the gaps exist that are going to prevent the field from moving forward. Most importantly now, you're going to point out where the holes are and how your research is going to advance that so that we can overcome that hurdle and fill in that gap of knowledge. So that is what you should be writing for the rigor of prior research. That used to be called premise. That's in the significance section. Okay, why? What you're asking here is why should this application be funded? Should this project move forward? Now, when we talk about the rigorous experimental design, here we're talking about in the approach section. All right, and so in the approach section, a rigorous design means you have the appropriate power calculations. You have, for every experiment, the correct amount of information for your reviewer to evaluate whether or not it's feasible. Do you need to blind the studies? Are you considering, I mean, all of these things are considered rigorous experimental design. Make sure that you have your authentication up to support that you are authenticating each of your steps. I have a side note here, include power calculations in the approach. A lot of people tend to put their power calculations in the vertebrate animal section. That is not going to be reviewed. If you do not include your power calculations within the approach section, then it's not going to be able, the reviewer has no way of evaluating whether or not you have scientifically considered the appropriate sample sizes. All right, that must be in the approach and not in the vertebrate animal section. Other things for a rigorous experimental design, you want to consider sex as a biological factor and other biological variables. And so when I'm talking about sex as a biological variable, if there are no known sex differences, then you have to power your study large enough to test to see if there are differences between the sexes. If there's a known sex difference, you must power it differently to be able to observe the differences in the sexes, right? And then if there's a reason why you don't need to consider both sexes, you need to justify that. For example, in the best I can give right now off the top of my head is if you're studying either prostate cancer or breast cancer, right, you're going to justify using just males or just females. So make sure that you're considering sex as a biological factor, you're powering accordingly. And then other biological factors, if you're using human samples, you might want to make sure you make it obvious to the reviewers what the age might be, what the race expanse might be, also the age. Are you looking across the lifespan? So if you have human samples, reviewers are going to want to know a little bit about the demographics, so make sure you include that as well. Again, authentication of key biological or chemical resources is important to know that you are checking to make sure that the chemicals you use are validated. Any cell lines you use are the cell lines that you're talking about. So that's an important part there, that you're authenticating each agent that you're using. Please, I want to highlight, please do not use the vertebrate animal section or the facility section for any additional experimental details. That's called overstuffing, and that can get you with your application withdrawn. A vertebrate animal section is not a place for you to start putting in additional rationale, and you should absolutely have no type of analysis in that vertebrate animal section. All you need to have is a brief description of the procedures, justification of the animals, and then what you're going to do to minimize pain and distress. That's it. No power calculations there. They belong in the approach. Same with facilities. A lot of people like to put in the facilities their exposure chambers or their exposure systems. Make sure there's no analysis in that aspect. If you're writing that up, that's another reason for withdrawal for overstuffing. In addition, NIH specific, do not put hyperlinks in there. Hyperlinks are also considered overstuffing. It's a way for you to take advantage and expand upon the page limit of your applications. Hyperlinks are not allowed. If you do include hyperlinks and they are egregious, it will get withdrawn. Also, reviewers we tell them, do not click on those hyperlinks. Number one, it could identify them. There could be malware associated. We don't know. Right. And so that could be a breach of confidentiality. And so we ask, please do not put hyperlinks in. There's only one place a hyperlink goes, and that's in your biosketch that links you to your NIH biosketch. It tells you all of your publications. That's the only hyperlink we should be seeing. Okay, so please do not overstuff your application. We do not want to have to withdraw just for that. Okay, so that's the NIH specific things you want to consider. Can I advance the slide or no? All right, whoop. Did we go too far? Is that the next slide? No, okay, there we go. Okay, so we went over the NIH specific interests and what you need to be aware of. We also have the CounterACT specific. And so remember, you have to have practicality in this real world civilian mass casualty situation. Once again, Shardell has already said this, but it has to be amenable for in the field or in hospital. So again, less than 15 to 30 minutes post exposure, your countermeasures should be working. Again, if it's opioids, that's another situation. Please contact your program official to just, talk about that. Again, the prophylactic therapeutic approaches that are administered before a chemical threat are considered non-responsive to the NOFO, and so that will not be considered for review. One thing we absolutely like to see is the repurposing of already approved FDA drugs, right? That's going to speed up the time to get it approved. It's also going to speed up the time to get it out there on the field. And so repurposing is a great way to go for this CounterACT program. Anything else, Shardell, you wanted to add there? If not, I'll just keep going. Okay. Okay, and then some more CounterACT specific things you want to consider. So in that research strategy, make sure you include the structure of your lead compound when possible. I do have medicinal chemists. I have a lot of people in that review panel who are very interested in seeing that structure. If the structure is not available or if you cannot include that structure, please provide an explanation. In your letters of support, right? You must include letters of support if you're mentioning collaborators. They need to see that those collaborators are there. Okay. Also, for those letters of support, some larger companies where you need to go to for their exposure for certain chemicals that you cannot do at your research facility. If you have a contract with a company, they should include a letter of support, but that letter of support should not have experimental details or any type of analysis. That should be in your approach, all right? The letter of support is saying that they are going to do what you ask of them in your approach. All right, so make sure you get that information into the approach and it's not in the letter of support. Again, that's considered overstuffing and could be a cause for withdrawal. Make sure you have your letters from your appropriate biosafety committees, et cetera. So not only must you have a statement saying you will handle the biohazards at your institute, right? And that's any of the chemicals, it could also be human samples, et cetera. You need to have a statement in your facilities or anywhere in like the resource sharing plan or in the specific agents. You can put it anywhere in the application. Make sure you address biohazards at your institute somewhere. In addition, if you're using one of those particularly nasty chemicals that needs approval from higher agencies, make sure you have that letter included as well. That would be in that letter of support. So that's two different ways you have to address biohazards, and that is scoreable. So please keep that in mind. And then intellectual property strategy, make sure you work closely with your institute, your tech transfer office, and you need to have that freedom to operate. All right, describe any known constraints to development and eventual commercialization of the products and how those issues could be addressed. And also, when applicable, discuss details of patents pertinent to therapy being developed under this application. Okay, we can go ahead and move forward. All right, other things to consider that the reviewers are going to be looking for: scientific feasibility, right? This in your approach, you talked about the significance of your research, right? How it's filling in that gap. That's the significance, and that's asking, should this application, be done? Now, when we get in further into that, your scientific feasibility here is Can your project be done and can it be done well? Right, that's what they're addressing. So make sure the preliminary data and the literature supports the hypothesis and the proposed studies. Remember, an R21 does not have to have preliminary data and it cannot be asked for. But of course, in the UG3, UH3, the preliminary data should absolutely be supportive of the hypothesis. So please, if you're studying chlorine inhalation, don't come in with preliminary data looking at sulfur mustard, something like that, right? You want your data to be supportive. Present sufficient details to evaluate the approach. Please do not just say, we're going to perform the analysis as we've done previously and cite 4 papers, right? Don't make it a challenge for the reviewer to go and try and figure out how your analysis is occurring. Put it in the application. Some of the reviewers are not going to go digging. All right, so put in those details, otherwise it's going to be insufficient to be able to justify feasibility. Make sure the work meets the goals of the projects, and that's where these milestones are key, right? The milestones are every year. And they're going to make sure what you're working towards meets those milestones. So if you go off studying chlorine exposure and you're trying to prove that this drug, you're trying to find a lead, right? Make sure that you're studying that and you're not going off studying a signal transduction pathway that's leading to that, right? You want that goal to meet that milestone to be able to move forward. If needed, make sure you're going to address those interdependence of aims. That's a bit tricky, right? Aims should always be independent of each other. But when you're looking at the UG3, UH3, you're taking one compound and moving it forward. So you might have to address the interdependence of some of those aims, and how that comes into play and how you can buffer that is by adding, which in the next bullet point says here, sufficient alternate approaches and research strategies. So, you need to have those alternate approaches there because you have very specific quantifiable milestones. If they're not going to be met, what are you going to do? Make sure that's there. And then again, if you have a team, which most people do, this is, all kind of different science coming in as one, make sure you have a well-written MPI plan. So this will show that the ability for collaboration. So if you don't previously have any publications together, the way to prove to the reviewer that you work well together is to put together a great application and a well-written MPI plan that tells you how you're going to communicate, when you're going to meet, how you're going to meet these milestones together, and how you move forward. Okay. That's it for me, I think. No? Next slide. There we go.
Sure, I'll just close this up. So thank you. So as Shardell mentioned earlier, CounterACT is a program that reaches across many institutes of NIH, each of them and their respective program officers with their contact emails are listed here. Jodie's contact information is here in case you have questions about the preparation of the application or some required documents. Now, if you have specific questions about how your research might fit with any of the ICs within the context of cooperative agreements or the R21 or the basic research funding opportunities that we didn't go over today, we encourage you to contact the relevant program officer directly. Once again, I will be sharing these slides with everyone, so you're going to have these names and contacts. And that brings us to the end of the presentation materials. I'll now turn to the questions. Please submit your questions through the chat box. I'll read them, and then Dr. Spriggs and Dr. Fleming will answer them. So far, there are no questions. So you might want to type quickly. With that, I'm gonna stop the share and we'll see.
I would say also, just if you need to unmute yourself and ask the question, don't,
Okay, sure.
Yeah.
Yeah, that's also fine. Okay, so I'll go ahead and read one question here that is coming through the chat, and that is, can you give an example of preliminary toxicity studies in UG3 versus tox studies appropriate for UH3?
Hi, I'm sorry. I was actively trying to read the second half of the question. So 2 parts of that. You don't necessarily have to do a lot of talks in the first part of the program. So what I think is that for the second half, you want to make sure that the dosing regimen that you're proposing isn't toxic. So even though the exposure is acute, sometimes you have to give the therapeutic more than once. So you want to make sure if it's IM and you are injecting it, that you don't have, the muscle tissue is okay, that, it doesn't have off-target effects on organs like the heart and things like that. So I think that depending on what you already know, the best idea would probably be to contact your specific program officer. And for me, it's best to have that specific aims page and look at what you're trying to do in UG3 and UH3. I've had to move studies around for people and add additional studies in to get them ready for the kind of tox things that we need to see to move forward, if that makes sense. So, I can see the earliest thing being the drug's not lethal. And then the second or later thing being that the mode that you're proposing to use it does not have toxicity and off-target effects, if that makes sense. I'm going to mute while this fire alarm continues. And I'll invite Dave, Claudia, Jodie, Neil, and any other program officers that are online to add to any of the answers.
Okay. All right. Well, Anjali, we'll come back to your second question in a minute. In the meantime, one question is, if you have a UO1 currently, are you eligible for the UG3 UH3 mechanism? And I mean, I can answer that. If you currently have a UO1, that's fine. That's great. You are allowed to apply, even while that is running, you are allowed to apply for a UG3 UH3 award. The research should be distinct from the one that you're currently being funded for on the UO1. Okay, so here's one question about those entry points to the UG3. So if the R21 grant is not sufficient for model development and target identification, then what steps does someone have in order to progress that to the UG3? Shardell, I don't know if you are able to come on to address that one. Okay, or, well, we can come back to it. In the meantime, Jodie, we'll go to you. So you mentioned that new therapeutic must show efficacy in 10 to 15 minutes post-exposure. Does this mean that I guess that we no longer support delayed effects such as status epilepticus that occurs days to weeks after an OP exposure?
Okay, so no, obviously that's important too. So what we're into, the primary thrust of the application, are we talking about, should be on that acute exposure and eliminating the, any mortality from that exposure. But of course, if we have cures available, right, or if you're proposing a cure and then you also want to look at the long-term effects such as seizures, et cetera, yes, that's perfectly acceptable. The main thrust should be looking at that one-time acute exposure, but of course we do support after that exposure occurs, increasing the quality of life of those individuals after. Hopefully that addresses that.
Okay.
Yeah. I see another one real quick. In the milestones, they don't have to be in the specific aims. They can be anywhere, most likely they're just going to be in the approach. And I do want to comment that the reviewers have a specific area on their review criterion where they note about your milestones. Are they quantitative? Are they sufficient? So that's a separate review criteria specific for the UG3, UH3 mechanism.
Okay, that's great.
If I could just add one more thing too. If for some reason for the R21s, your drug or your target is not hitting that 10 to 15 minute post-exposure right now, right? As long as you justify that moving forward, your goal is going to be to optimize it to meet that time frame. That's a great entry point too. So it's or if it's right now it's IV drug, which is not good for the field. Please, you can continue with those studies with the emphasis and full well knowing to the reviewers that you're going to ultimately work on that to make it a better delivery system, whether it's IM, inhalation, etc.
Okay, yeah, that's very helpful. Thanks. Okay, another question. So if you have an FDA approved drug already that has tox data, but it's via it's tox data from a different route of administration. So is it appropriate in the UG3, UH3 mechanism? Is it appropriate to propose tox studies that are going to be done from the expected route of administration?
I think that would have to be a control, right? It might have to be a control depending. That's going to be very specific as to how you're writing your approach, I think. Yeah. That's really going to be in your own individual experimental design, depending upon the drug itself and what you're proposing and how to change it. So you would want to meet with your program officer about that, for sure.
I'm sorry, I'm going through. Did Chuck got his answer about long-term effects? Okay, just let me know specifically if I need to chime in so I can
Attention, please.
keep this.
There has been
Yeah.
a fire.
Okay. So, okay, now I'm trying to read this question. Let me see if I can phrase it. Okay. Neera, I don't know. So I'm reading your comment in the chat about
This
some
is
of the activities are expensive. Do you want to unmute yourself and ask your question?
Yes, so Neil, thank you so much. So this is following my first question that I had asked. So please let us know if the R21 grant is not sufficient for the model development and target identification. What are the steps to progress this to UG3, number one? And this is because some of the agents that we can only use at the contract labs to do the model development as well as screen the drugs is going to be very difficult under one R21. So this is something that we don't know how to, address at this point.
So Neera, I would urge you to send us a specific email, because that's something that, depending on the model, like that's something that maybe Shri or Alexi may have to weigh in on, and then they may be able to work with CCRP at NIAID for bridge funds or model development activities if it's not enough to do it here, or there could be some kind of where you just do the preliminary development in the R21 and then maybe fine tune some things in the UG3 phase of it as opposed to the UH3, and then it wouldn't get kicked out. So it's just, you can't come in brand new and say, I'm going to develop an animal model in the UG3. But if it's building on R21 work and it's just, some fine tuning that needs to be done, then I don't see why that would be a huge problem. Like we say, each toxidrome is specific and it has different needs and different bodies of evidence out there. So if you email us specifically, then we can, try to see and get those needs met.
Yes, because with this new mechanism, we're really finding, especially when I've been going through all this and we've been there before. So it's something which is a little difficult to understand, and that's why I was here. But thanks so much for giving so much information. But still, I feel that the R21, the amount, the way things are going, because we are doing studies at the contract labs, and we've seen changes in the past 2-3 years, and how things have been running. Believe me, we had to cut more than half our studies, and that doesn't make sense. We're spending money, but we don't want that money to get wasted, then we don't get the meaningful data. And that's what I've been trying to stress a little bit, that because things have gone up, the R21 at this point gets a little bit, not sufficient and not enough, time to get this whole thing done to go directly to the UG3 phase, because sometimes there are those mechanistic analyses that now come under basic ones, and we don't have anything for skin anyways right now. But for the other things, because I work majorly with skin and eye, so that part piece is right now missing. And the third thing what I'm noticing is that this transition, it's good, but maybe not enough to jump to the UG3 phase as of now, because if you can't have your already identified, drugs there, or, a model set up, then you can't jump to UG3, and maybe R21 will not be sufficient to do that if the agent is something that we have to do in the contract labs.
Well, if I could just say one thing here, one of the activities in that UG3 phase is developing the relevant post-model exposure animal models, right? So if you use the R21 to get those targets, or narrow down your targets, that's what you're looking for, right? You want to come in with a lead target, a candidate. Then you're going to start, then that's when you're going to bring in your big contracting companies and actually develop that model system. That is a definition of one of the aspects to be in the UG3 phase. So that's where the larger amounts of money should come from, is in that UG3. So spend that time and effort on the R21, getting that lead candidate as the entry point.
So how do we get the lead candidates if we don't have a relevant? Do you think we do it in cell culture or whatever else, then coming to the in vivo models, because we do need a model and the biomarkers to test the compound? So see, this is the catch-22.
No. So for the purposes of this webinar, I think we have to move on. But Neera, I so appreciate your comments, and I want to continue this conversation just offline so we can talk about it. Okay.
Thank you.
No, I appreciate your comments very much, and it's very relevant.
Thank you.
Okay. We have a couple of questions that we haven't yet gotten to, so I'll go through them. There's one that says, I saw that nerve agents and pesticides are in different toxidromes. So are underexplored nerve agent antidote therapeutics, are these candidates for exploration under the pesticide UG3, UH3?
Okay, so all the toxidromes are candidates under the UG3, UH3. It just matters. We're just grouping them. So you're going to have your anticholinergics, and then you're going to have your convulsants, if that's what you mean about how we're bucketing them. But both are eligible, all the toxidromes. The two NOFOs we talked about today cut across the entire CounterACT program. And so any chemical threat that is on that list is eligible. And what's more important is we don't rank them in priority when you're coming in for these. So a convulsant is not going to have higher priority over a cholinergic because it's understudied, but we could have interest in that. And we are interested, of course, in expanding portfolios to understudied chemicals, but for the purposes of review, we don't rank the chemical threats that are studied. Once we get our scores back, we consider the scores and we consider portfolio balance and funding but it still has to be a good application and come through review and get a good score.
Okay, so Lindsay, hopefully that answers your question. Otherwise, feel free to unmute yourself and restate it. Okay. In the meantime, so I think the last question that we have is for PK studies that are in the UG3 phase, is that with the chemical threat on board or without?
Yeah, so if you go to the UG3 NOFO, you'll see that one of the last things are just demonstration of preliminary safety PD/PK properties of the candidate therapeutic. So that can, just be an IV injection just to get that 100% and see what the distribution is or something like that. But we can talk offline for specific studies, but we do want to see PK.
Yeah, and then that's specifically on your therapeutic. You do not at that point in time have to have the chemical threat on board. You want to first prove that your antidote or your treatment is safe, right? And so that's without the chemical threat.
Yeah, and thanks if I didn't clarify that too. Yeah.
Okay. Those are all the questions that we've had submitted. If anyone has any remaining questions, you can unmute yourself and ask them. Otherwise,
Did we address the question from Oliver about for the R21 submission?
No, I did not address that question. Or I did not ask that question. Let's do that. Okay, for the R21 submission to PAR 23-139 in May 2024, can we read that creation, validation of clinically relevant in vivo models of post-exposure, does that allow for applications describing systems and molecular assays to gather biological material and analyze it to elevate injury, to evaluate injury?
Okay, so for the R21, it has to be in the interest of developing a therapeutic. So I guess, and I hope that, and that's a distinction that the PI is going to have to make in their application. So there are some basic NOFOs out there where all you have to do is deliver the chemical threat, and then you would gather biological samples and research the pathology, if that makes sense, with either standard of care on board, but usually there's no therapeutic involved in the basic NOFO. So if you're talking about no therapeutic development or with nothing in mind for that, then that's out of scope. But there are ways to do this with the thought moving towards a therapeutic. And so that would be the argument that you would have to make. And I would urge you, there's ways to do that. So I would say, please contact the scientific contact that's listed for your toxidrome, and they can help you with that and help draw those distinctions clearer.
Because otherwise that might be what, your R01?
It would be the R01 if all you want to do is to study the pathology of disease after chemical threat exposure. That would be the R01, and that's kind of what some of them are for, and some of them are looking for mixtures of compounds. So you have to look at more than one for commonality in the pathology. So the R01s are very tailored, and you have to look at those individually. But this question is something that can be posed and is exactly the type of questions that should go into an email to the scientific program contact. I want to do this. which NOFO do I apply to? And that's exactly what we're here for, is to say, hey, if you take out this study, you can go here. If you add this study, you can go there, but make sure this is your end goal and you're doing also XYZ. Like that's exactly the emails that we hope to receive and can really help you develop responsive applications.
Okay, I think there's one last comment about for the skin related studies. There's no R01. I believe that there's an R34 mechanism that they use to support some of that NIAMS will use to support some of the basic research for skin. Okay, with that, I think it brings us to a close of this webinar. I'd like to thank you for your attention. I'd like to thank Jodie and Shardell for everything that they did to present at this webinar, all the way through, persisting through the fire drills. And to everyone, we'll make the recordings and the slides available, and you'll get an email when they are ready. Okay, thank you, everyone.
I have one thing, Neil, I'm sorry, because I got a direct chat. You can also, if you email me and tell me what chemical threat is, I can tell you who the scientific contact is if you're unsure. You can email me, you can email Neil, and we can get you to your scientific contact, okay? If you don't know how to, to read it in a NOFO or aren't sure, we're also here for that. Sorry, I agree with that, that's it.